pratiche. Qualcuno ha chiesto di poter visitare il Palazzo Ducale. È possibile organizzare una visita? Chi è interessato si presenti, lo dica in segreteria. Dall'1 alle 2 possiamo organizzare un gruppo per chi è interessato. Qualcuno mi ha chiesto del libro degli atti. Gli abstract saranno pubblicati online. Tutti coloro che sono iscritti al convegno e che hanno mandato un abstract riceveranno un'informazione via email quando saranno disponibili e rimarranno comunque sempre disponibili sul sito della SIB e sul sito del congresso. Chi ha presentazioni nella seconda parte della mattinata, anche nell'altra sala dove lavoreremo dalle 11 in poi su 2 sale in parallelo, è pregato di consegnarle allo stesso centro slide che avete visto di fronte alla segreteria. E adesso buon lavoro. Maurizio.
La visita al Palazzo Ducale comprende la visita alla galleria?
Sì,
o no?
sì. Comprende la visita alla Galleria Nazionale delle Marche. Bene, buon lavoro, grazie a tutti. Menico e Luisa.
Grazie, quindi siamo pronti per iniziare e ho dato i 2 secondi di tempo. So welcome everyone, it's really a pleasure to have you here. When Luisa, we organized a session entitled Frontiers in Structural Biology, we did realize it might be over-ambitious, but anyway, we thought even in a session of 2 hours, we might have an idea of what is going on in structural biology in the frontiers nowadays. And that's, I think, is the major reason we decided to invite as a keynote speaker Professor Wolfgang Baumeister, which is really an honor and a pleasure to have you here. Thank you very much for accepting our invitation. And Professor Baumeister is the director of the Department of Molecular Structural Biology at the Max Planck Institute for Biochemistry in Munich, in Germany. And of course, where a number of groups are studying really challenging supermolecular assembly using cutting-edge technology like cryo-electron tomography, visual proteomics, phase contrast methods, amongst others. And I think he's really a top person in the field of modern structural biology, and to be in a position to really use multidisciplinary approach to solve, from a structural biology point of view, really challenging problems. And his research activity is actually, I would say, focused on the structural function relationships of supramolecular assemblies, involving mainly protein quality control, like, of course, the proteasome, the ribosomal supercomplexes, but also synaptic complexes and other digital complexes, tripeptidyl peptidase, and so far, so on. So he's the recipient of a number of, of course, awards and recognition. I don't think it's really important to mention all of them. He received medals and prizes from Germany, from Israel, from Italy, the medal from Padua, and is the editor-in-chief of the Journal of Structural Biology, member of the editorial board of many journals, and I think he's really the person to give us he's really the person to give us, so you didn't hear anything of what I said. I'm very sorry. I said very good things. So it's really a pleasure to have you here today and to deliver the first talk, which is entitled Structural Studies on the 20S Proteasome ex situ and in situ. Thank you very much. Sorry again for the mistake in the microphone.
Okay, thank you very much. Thank you very much for the kind introduction, and thank you for inviting me to Urbino. So in the next half hour, I will talk about structural studies of the 26S proteasome. In the first part, I will talk about studies of the 26S proteasome ex situ using basically the established methods of structural biology, cryo-EM and X-ray crystallography, often in a kind of a hybrid approach. And in the second part, I will talk about structural studies of the 26S proteasome in its functional cellular context, and that is done by electron tomography, a method which we have developed over the past 20 or 25 years. Now, let me begin with a very simplistic representation of the main eukaryotic pathway for intracellular protein degradation. And the key player in that is, in fact, the 26S proteasome. It's a molecular machine of 2.5 megadalton. It degrades, proteins marked for destruction by the covalent attachment of ubiquitin or polyubiquitin chains in an ATP-dependent manner. And it generates peptides, which are typically 8, 10, 12 residues long, which are of very little use for the cell, with one notable exception, and that is the immune system. But even in the context of the immune system, the peptides generated by the proteasome usually need quite a bit of trimming in order to fit the MHC complex. Now, but the majority of peptides generated by the proteasome has to be degraded further. And there is another very large molecular machine, tripeptidyl peptidase. In fact, it's about two times as large as the 26S proteasome, a 6 megadalton complex. But unlike the 26S proteasome, it's built from only one type of polypeptide, so it's quite a bit simpler in composition. So what it does is basically it cleaves off tripeptides, which are then further degraded to complete the catabolic pathway by an array of aminopeptidases generating free amino acids. So what I will do in the course of my talk, I will, very briefly talk about TPP2, and I will do that in order to sort of illustrate a relatively simple approach, integrative approach. But also I will do it very briefly to highlight some of the key features, the hallmarks of intracellular proteases. And then but the main part will be then on the 26S proteasome. So TPP2, that is a low-resolution structure obtained by cryo-EM almost 10 years ago. It's basically, it comprises two twisted strands which make contact only at the terminal elements here. And you can see these strands are kind of segmented, so each strand comprises something like 10 segments, but that is somewhat variable from species to species. In humans, it's only 9. This is the Drosophila structure. And then, you can simply, as you divide, the total molecular mass of almost 6 megadalton by 20 segments, you see that each segment must be a dimer of roughly 1.5 kilodalton monomer. So we tried at the time to solve the structure of TPP2 by X-ray crystallography, but that was not very successful, and the reason for that is illustrated here. The structure, as I just showed it, this 6 megadalton complex, exists basically only in a very limited concentration range here. But that concentration range, coincides, with the typical protein concentration in the crowded environment of the cell. As you increase concentration, the two strands of TPP2, they begin to grow, and you generate quite a degree of polymorphism, which is obviously not conducive to crystallization. As you lower concentration, the thing begins to disassemble here, and you can, do that in a fairly controlled manner, generating either dimers or tetramers. Now, as you then try to crystallize those, of course, you go into this direction again, and you end up somewhere here. So what we tried to do at the time was that we tried to modify the dimers and the tetramers by various treatments, by fusions, by mutations, by chemical modification. All that did not work. What did work was, exposure to a mild detergent octyl glucoside. In the presence of when some octyl glucoside binds to the dimer or tetramer, then, they remain dimers or tetramers as you increase concentration. So we ended up with the structure. This is the structure of the monomer. It shows its domain organization. In yellow, you see, the catalytic domain. It's very, very closely related to cytolysine, but it has a long insertion shown in orange here, which forms a separate domain. Then we have in green what we call the central domain, and then we have the C-terminal part, which is completely alpha helical, and it's a 10 helix bundle, in the monomer, and as it assembles into a dimer, it forms a 20 helix bundle, and that 20 helix bundle essentially determines the curvature of those strands here. So, for instance, in humans, where we have one helix less, there's a little bit more curvature, and as a consequence, we have only 9 segments instead of 10. So what we have done here is basically we have fitted the 300 kilodalton dimer structure into the then existing EM map in order to obtain a hybrid structure of, so you see in blue again, the C-terminal domain, which, as I said, determines the curvature. Now, later we did then human TPP2, which is a little bit shorter, as I said. You see here again the structure of the dimer. So in yellow, that is the cytolysine domain with the active sites highlighted here. Then this intermediate or intervening domain, and then the central domain and the C-terminal domain. Now, we generated in this case a pseudoatomic model of TPP2. Now, as I said, I wanted to highlight a few features typical for all intracellular proteases. And of course, intracellular proteolysis is a dangerous thing, and you have somehow to protect, cellular proteins from sort of random attacks by a protease. So the hallmark of all these proteases is that the active sites are sequestered in what we call a catalytic chamber, which is buried deep inside these strands here. And that is, and the active site, I should say, is not active in the dimer state. It becomes active in the tetramer. So it's a contact-induced activation of the active site. So the important thing is that activation follows sequestration of the active site in this catalytic chamber here. So any substrate to be degraded has to pass this entrance left and right of this helix bundle, go into the so-called foyer, and then we come to the antechamber. The antechamber has a width or a diameter here of something like 10 angstroms, and so only unfolded protein or unfolded, polypeptides or oligopeptides can enter, the catalytic chamber here. No folded protein, can be exposed to the catalytic site. So much for TPP2. Now, the remainder of my talk, I will talk about the 26S proteasome. It comprises 2 subcomplexes. One is the 20S core complex, that is where proteolysis takes place. And again, it's a cavity here formed by the beta subunits where proteolysis takes place. So any substrate to be degraded has somehow to enter, this core complex and end up in this central cavity. The alpha subunits form a gate that controls access to the core complex. Now, the 20S proteasome in eukaryotes is not, in a similar way also in archaea, where we have a simpler form of the 20S proteasome, but the same basic architecture nevertheless. So it's not functional without, associating with this regulatory complex, the 19S caps. Their role is basically to prepare substrates for degradation, to recognize ubiquitinated proteins, to deubiquitinate them, to unfold them, and to help them in their translocation into the core complex. Now, the core complex, we determined a long time ago by, let me say here, that is, that is an old picture of the 26S. For many years it was, the textbook image, because, it took 20 years from moving from the structure of the core complex to the structure of the entire 26 complex. So the core complex we determined a long time ago by X-ray crystallography. I don't need to talk about that. Let me mention just that we're at the challenge today to solve something like the 20S proteasome. We would not do it by X-ray crystallography. This was, years of crystallization trials This is an EM structure at a slightly higher resolution, as a matter of fact, and that was data collection in 1 day and the processing in 2 days. So this is quite a difference. But the 20S is relatively easy compared to the 26S proteasome, as you will see in a minute. So, and as a matter of fact, this is not the limit. We did not push the envelope here. We have, in the meantime, using some newer technology, gone to 2.6 or so, and there's no reason that one could not push it to even higher resolution. Now, well, I think, again, that is history. I think we don't need to mention that, but just for the sake of completeness, the crystal structure at the time, in conjunction with, mutagenesis, revealed the very unusual active site, a single residue threonine, in 3 of the core subunits. So the talk today will be mainly about the 26S proteasome, and I think this is just to sort of introduce the way we approached the structure. The main element was an electron density map at medium resolution at the time, as you will see, something like 6 angstroms, in parts better than that. And that was interpreted in the light of data or of, let's say, experimental constraints or orthogonal data, which mainly came from cross-linking and mass spectrometry. Now, why was it so much more difficult to determine the 26S structure compared to the 20S? First of all, it's the sheer complexity. In a typical 26S proteasome, there are 34 canonical subunits plus 6, 7, or 8 associated proteasome-associated proteins, which are usually present in substoichiometric amounts. So there are, first of all, the core particles, 7 alpha, 7 beta subunits, of which 3 have the active site. Then, most importantly, there's a whole module formed by AAA ATPases, which have non-redundant functions. They are very similar, but nevertheless, some are involved in substrate unfolding, others are involved in substrate translocation. Then, there are 2 resident ubiquitinous receptors in addition to a number of shuttling ubiquitin receptors I will mention later on. This is RPN10 and RPN13. There's a little peptide here which has an important function in assembly, so it's a kind of an assembly chaperone, also stabilizing another module of the whole thing. Then there are the 2 MPN domain containing subunits, RPN8 and RPN11. Particularly important is RPN11, which is the only resident deubiquitilating activity, and again there are associated ones, which are not canonical subunits. Then we have the PCI module containing subunits. They serve basically a structural role, as you will see. They enable collective movements, which are important in order to, in transactions of substrates. And then there are the 2 largest subunits, RPN1 and RPN2. They are extremely variable, extremely flexible, as you will see. They basically serve as hubs for the association of proteasome interacting proteins. Now, but the main problem is not complexity. The main problem is heterogeneity, and there are 2 sources of heterogeneity. One is the fact that not only some of the proteasome interacting proteins, but also some of the resident ones are present in substoichiometric amounts, so there's compositional heterogeneity, but in addition to that, there's enormous conformational heterogeneity. So what you see here is a low-resolution structure, and in orange superimposed is a variance map. The variance map highlights where the variations are, and they are all, all the conformational heterogeneity is in the regulatory particle. You see, basically, there's very little dynamics in the core particle. That is the reason why it was relatively easy to obtain crystals of the core particle. Now, the basis for the structure determination was at the time a 6 angstrom map of the 26S proteasome. As the color changes, it indicates that, a global number for resolution is relatively meaningless, where it's blue here, and that is the core particle plus, the ATPase module. Resolution is quite a bit higher, and what you see in red, these are the hotspots of variation. So the integrative approach we have taken is basically illustrated here. What you see in the top row is, I mean, the ingredients of the modeling. It's the EM map. Then we determined a couple of positions of subunits from EM difference maps. So we created, I mean, difference maps between the wild type and knockouts, especially of the two ubiquitin receptors, RPN10 and RPN13. So in green, you see the difference densities. So we had the absolute positions of some of the subunits. Then, as I mentioned, I mean, we did a lot of cross-linking at mass spec in order to come up with an interaction map. And so basically, we tried to map this interaction map onto the electron density. And then we obtained, I mean, crystal structures of a number of subunits in order to, to be able for some of the homologs to obtain, I mean, homology models, but also, I mean, to fit them into the final map, as you will see. Now, the modeling went the following way. Basically, we had a sort of a beat representation of the subunits or domain, and all these beats were placed in the boundaries of the EM density map. Then they were jiggled around, and each and every configuration then was scored against the violation of experimental restraints coming from cross-linking and mass spec. So that provided us, I mean, as you will see in the next slide then, with the approximate positions of all the subunits and all the domains in the case of multi-domain subunits. That was then refined by flexible fitting based on molecular dynamics to improve the quality of the fit. So we evaluated a total of 1.6 million different configurations for the subunits. Of these 1.6 million, there were only 4 violating either none or at most one of the experimental restraints. So in other words, there was an almost unique solution with the exception of RPN11 and RPN8, the two MPN subunits, close homologs, which form a heterodimer in the complex. Otherwise, I mean, we had an almost unique solution. So the basic architecture was the following. RPN1 and RPN2, these flexible hubs, I mean, are located at the distal end, or I mean at the site of the AAA ATPase module, which we knew already at the time was directly docked onto the alpha subunits of the core complex. Then the two ubiquitin receptors are again at the distal end. The deubiquitinating module with RPN11 is sitting here, basically near the top of the AAA ATPase. And the PCI subunits here, they form this horseshoe-shaped, I mean, framework surrounding basically this MPN module here. Now, for some parts, we could already at that time, I mean, put forward an atomic model because we had determined the structure of RPN6, for instance, which has a PCI module, and that PCI module was pretty clear that it formed this, I mean, the contacts in this horseshoe-typed arrangement. That was what we observed in the crystal packing. 6, I mean, of these subunits formed this horseshoe. And so we expected that also, I mean, in the heterohexamer would form a similar configuration, and we observed at the time we had a lower resolution map only, a kind of a horseshoe arrangement here in that part of the molecule. So, but we checked that, I mean, more rigorously, for instance, by, I mean, doing a low-pass filtering of RPN6, cross-correlated it with the entire electron density map. And so in each of the regulatory particles, there was one very well-defined cross-correlation peak, indicating the position which was in agreement with the model. Now, I mean, as resolution improved, we were able basically to put forward atomic models of most of the subunits, because this was facilitated by the fact that basically almost all the subunits are completely alpha helical. So I mean, at 6 or slightly better resolution, I mean, we could, I mean, interpret the map, given, I mean, the other information we had, with the exception of 2, RPN8 and RPN11. These are the only 2 subunits which have appreciable amounts of beta sheets. So we went back to X-ray crystallography in this case. And and we obtained crystal structures of a recombinant RPN8, RPN11 heterodimer. The interesting feature of this heterodimer was, again, that, I mean, as such, it was enzymatically inactive because there's an insertion, this loop here, that hinders access to the active site zinc molecule here. And that, I mean, inhibition, autoinhibition is relieved when the whole thing is integrated into the complex. So we had a 1.8 angstrom map of that one, which we could integrate then into the electron density map. So I think this is a short movie, which is not meant to illustrate the assembly pathway. It's meant to illustrate basically the architecture. So we disassemble everything and then rebuild it brick by brick. So we begin, I mean, this is the platform of the alpha subunits here. Now come in the 6 AAA ATPases. Some of them have a C-terminal tail, which inserts into a pocket here. And insertion into the pocket then triggers opening of the gate in the center of the alpha ring here. In comes then RPN1 and RPN2, these hubs. RPN2 is relatively well defined. RPN1 is poorly defined here, very low resolution. As you will see later on, it's extremely flexible when we disentangle the coexisting conformations. Then come in all the PCI subunits forming that horseshoe, and they sort of enframe RPN11 and RPN8, the ubiquitinating module. And finally come in the 2 resident ubiquitin receptors sitting at the far end. So probably they were added late in the evolution of the complex. So this is, I mean, the basic architecture. And in the next, well, in the meantime, there have been a couple of technology developments. I should say that the most important one is automated data collection, so we can operate microscopes now 24 hours a day, 7 days a week, and so typically generate something like 1,000 images per day, which is a lot of data. But that enabled us to collect a data set of 5 million individual particles for, I mean, well, disentangling coexisting conformational states. So there are other developments like improved detectors which play a role, but the data I'm showing today, they were still collected with the old CCD cameras. But I mean, we are collecting a large data set, I will mention that now with also with the new detectors. So that enabled us, and I will again summarize that only in a brief video, to disentangle coexisting conformations. So basically, I mean a hierarchical classification of a state 1, a state 2, which is less populated, and a state 4. So these were the different conformations, and then, I mean, we could separate them, and you see where the dynamics is. So this is RPN1, for instance, which exists in a large number of different conformations. Then we have large movements here, I mean, of the PCI. Well, we have first a shift of the AAA ATPase module, which is particularly important as we move from S1 to S2 and S3. Then we have a rotation of the horseshoe, and with it of the MPN subunits here, a rotation of something like 22 degrees, which is so pretty large. And then that is followed as we move from S2 to S3 by another shift of this entire module. I mean, the PCI and the MPN containing subunits, they all insert into a very large 8 helical bundle, and that, I mean, being bundled there enables them to move collectively in a coordinated manner. I think the purpose of all that is illustrated in a little model here. Initially, the 2 ubiquitin receptors bind tetraubiquitin. We know where the substrate is, and the substrate has to be brought above the mouth, positioned above the mouth of the AAA ATPase. And then also the MPN-containing subunit, this one here, has to be positioned such that it can cleave off the tetraubiquitin, which is a prerequisite for unfolding by pushing it or pulling it here through the AAA ATPase channel. So I think an important step in the conformational cycle is between S1 and S2, that is the S2 state, well, sorry, that is the S1 state, this is the S2 state, and I mean, one can sort of capture this state here by the addition of ATP gamma S, and as you will see here now, the unfolding channel and the translocation channel are aligned, whereas in the case of S1, they are misaligned and any substrate would end up, I mean, here at a dead end. Now, okay, I think we are currently, this is ongoing work, we have now substrates which we can stall in transit, and we hope that we can arrest in that way, I mean, one conformation, and so we are collecting a data set now with the new detectors, but again, we have to probably collect a data set of a couple of millions, which is likely to give us, we can predict from the existing data a resolution probably of 4.3 or 4.5 angstrom. So we might also discover a couple of new conformations. I think the conformational landscape is not fully explored. I think in the last couple of minutes, very briefly, some electron cryotomography. I think I'm running a little bit late. So basically, this enables us to study structures like the proteasome in the context of cells. It combines the best structural preservation we can achieve, or that is physically possible, with the power of three-dimensional imaging. Well, I think I can skip that. So we have studied 26S proteasome in neurons. Neurons we grow directly on an EM grid. So that is an EM grid, there's the carbon film, and here are the neurons. Now, these studies were made possible by another technical development in the lab, and that is the so-called faceplate. I mean, normally in EM, phase contrast is generated by defocusing, and that requires very complicated post-processing, correcting for contrast transfer function. The faceplate generates pretty good phase contrast in focus, so the images are directly interpretable. So that was developed by Rado in the lab and is now commercially available. Now, I think that is now, we zoom somewhere into the cytosol of a hippocampal neuron. And at a glance, I think it looks pretty non-interesting, I would say, but as you zoom in and look at this guy here, so these are slices through a tomogram of an individual 26S proteasome deep inside this neuron. Now, we do the same thing here, and those at least sitting in the front might be able to recognize basically all the features of the 26S proteasome. There's no averaging yet. This is just the individual 26S in situ. And so what we can do is obviously, well, we can extract now a few thousand from subtomograms, but that is a data set of more than 100 tomograms, which takes weeks or months of data collection. We use some template matching, so we just have a template, which is a little cylinder with the right length, and search then the tomograms for the positions of these 26S proteasomes, and of course they occur in random orientations in the cell. So I mean there are a few where we look, I mean, perpendicular to the long axis of the molecule. There are others, I mean, indicated here where we look basically down the long axis. So we can extract all these tomograms now from, or subtomograms from the data set. And we have done that currently with about 2,000. We are analyzing currently from a different organism a data set of something like 10,000. Now, then we can, of course, create an average, and there was a surprise when we did an average of the global average of the 2,000. That one regulatory complex was much more prominent than the other one, indicating that that was not present in stoichiometric amounts. So as we calculated a variance map, it was clear that the main variation in the data set was the presence or absence of one regulatory particle. That was a surprise because, but I mean, it's sort of misleading in biochemistry. Everybody tried to optimize the yield of double-capped 26S proteasomes. And in the other one, we had some distinct variations here, which we could analyze further. So the first main difference amongst the particles is that only something like 1/4 of the particles has regulatory particles at both ends, and only about 3/4 is sort of single-capped. We could pushed the analysis quite a bit further by now analyzing, the one regulatory particle at one end here, and these are again in the variance map, and we see that, the variances are relatively well defined. And so when we separated them based on the tomography data here, we observed two states, roughly, 30% or so is in, and I will explain that in a minute, in what we call the commitment state. So it's engaged in dealing with substrate, whereas the majority under the conditions of the experiment, in the absence of proteotoxic stress, is only in the ground state. It's sitting there and essentially waiting for substrate. Now, when we take our high-resolution structure, from the single particle experiments here, and morph the two conformational states, we can compare them with what we see in the tomograms. And that is where we can conclude from which one is in the ground state and which one is in the commitment state. Well, and yeah, this is docking the high-resolution structures into the two different states. Now we can deal with, let's say, the 1/3 that is under the conditions of the experiment in the commitment state, and then we can analyze further this extra density here, which is highlighted in red. So this must correspond, and it's sitting at the right position, as we know, to substrate. But the surprising fact is that the volume we see here is quite a bit larger than the volume of a typical substrate of the proteasome. So I think there must be other factors associating with substrate that is in the state of being processed, which is not surprising. So basically, this is what we call a molecular census of the whole system. So as I said before, roughly 3/4 are single capped and only 1/4 is double capped. Then it's a minority that is in the commitment state, so the majority is not dealing with substrate under the conditions of the experiment. Of those in the double-capped ones, again, roughly half is not, in a conformation which we would conclude is proteolytically active. And then, the majority there, has activity going on only at one end. That is a very small minority. Now, I think the nice thing is that for each and every molecule we analyze, we keep very precise information about its cellular location. So we can say this one here is, single capped and dealing with substrate and so on and so forth. Now we are doing that currently systematically, but in Chlamydomonas, and as a matter of fact, it's a little bit misleading because the majority of protein degradation by the 26S proteasome happens in the nucleus, relatively close to the inner nuclear envelope. So we ignore that in that experiment here. So, yeah, basically, I think this summarizes what we can do in situ is we can determine the position of each and every 26S proteasome. As a matter of fact, with a precision of roughly 1 nanometer, we can, for each and every molecule, say what its state of assembly is, and we can infer from its conformational state whether it's dealing with substrate or whether it's sort of silent. So I think this is currently all at a resolution of roughly 22 angstroms. We are quite hopeful that also in situ we can push resolution, and we have shown that for ribosomes, for instance, but ribosomes are easier to deal with than proteasomes for a couple of reasons. They have higher contrast, but we think we can push it to 10 angstroms also in situ. So that brings me to the end. So that is the group of people in my lab working on the 26S proteasome. The in situ work is done work by Sho Asano and Yoshi Fukuda. Florian and Stefan, together with Friedrich Förster, have done most of the high-resolution single particle work. Andreas Brache and Ganesh Patar, who is now in Basel, have done the X-ray crystallography. Beate, together with Anne-Marie Schönegger, is TPP2 Eri rejoined the group recently. She has done all the knockouts for the absolute mapping. Pavel has done the work with ATP gamma S, and Pia and Elizabeth now at UCSD. They have done, I mean, the flexible docking based on molecular dynamics. So the integrative approach was a collaboration with Andrei Charlie's lab at UCSF. We used some help with knockouts from Keiji Tanaka. The mass spec cross-linking was a collaboration with Rudi Ebersold's lab at the ETH in Zurich. And I thank you for your attention.
So, thank you very much. I think it's really, really impressive. I wonder if in the future we go to in vivo structural biology at certain points. So certainly the open for discussion and question. Yeah, Martino, there's one there.
Thanks for your presentation. It was very enthusiastic and enjoyable. I didn't understand exactly the pore in the 20S subunit, so surrounded by the alpha subunits, leading to the death chamber, is opened by the interaction with the AAA ATPases only, or you require hydrolysis of ATP each time it is hydrolyzed and it opens it?
No, it's normally in a closed state, and that is why, I mean, basically isolated eukaryotic 20S have no activity. So, I mean, in the past, what people did was, I mean, they sort of artificially induced some sort of gate opening, for instance, by adding some detergent. The detergent, I mean, sort of disordered basically the structures that closed the gate. But that is, of course, a completely non-physiological situation. So I think every 20S proteasome needs a AAA ATPase to open it up under physiological conditions. In archaea, these are homohexamers of AAA ATPases. I mean, there's this called PAN, proteasome activating nucleotidase, which is relatively simple. It does not require other factors, but I mean, the AAA ATPase is always required for physiological gate opening. The remainder, I mean, is basically to position the ubiquitin receptors and then, I mean, translocating in a fairly complex way the substrate to the AAA ATPase. That is basically the story there.
Is there, yeah, Professor Bonori, before we...
I have two short questions. One is, now that you can count the proteasomes, how many single proteasomes there are in a neuron cell? The number, how many?
How many in a cell?
Yeah, you can count them now.
Oh,
100,000, 10,000?
No, several thousand.
Several thousand.
I think we don't have exact numbers yet because, as I said, for technical reasons, we could until recently not include the nucleus in our analysis because the nucleus is simply too thick and not electron transparent. So, I mean, that was the reason why we worked initially in these experiments with neurons, because most of the neurons have, I mean, are thin enough in order to be transparent, but we have now developed other technology to target any structure in the cell. So we use focused ion beam milling under cryo conditions to cut thin lamella. Now we can study them in the nucleus. I would say we can say from a typical Chlamydomonas nucleus, which we use because it's a little bit less crowded than other nuclei. It's less dense. I would say that the nucleus alone is inhabited by maybe 3,000 to 5,000 26S. Many of them are in very close contact with the inner nuclear membrane, so they have also distinct densities, I mean, linking them to the inner nuclear membrane. And it is my suspicion that much of the famous ERAT system, where I mean proteins in the ER have to be retro-transported into the cytosol, that is the model, in order to be degraded. Much of that pathway happens, as a matter of fact, well, the ER is of course continuous with the inner nuclear membrane, I mean, is executed in the nucleus. We knew, I mean, they have nuclear localization signals. We know since a long time that, and Ulrich Hartl in our institute has shown that probably 60–70% of the intracellular proteolysis is carried out in the nucleus. So, I mean, from our current data, I can't give you a number, but I think probably saying that it's 5,000 to 6,000 or so is roughly right.
That was the short question, actually. But I have another question. Now that you have shown that in the cell, I mean, the 2/3 of the proteasomes have only one cap on, and the other cap, it's not strange at being so mobile. Maybe they can be flexible. I mean, these conformational changes are very large, but they're still probably milliseconds or whatever. The question is, how fast is the freezing of the sample?
Well, I think the question was usually asked a long time ago in a different way. I mean, people were puzzled that substrate was fed into the proteasome from both ends, and that could be a little bit problematic. It's also still not clear how substrate is released from the proteasome. There's a little bit of NMR work from Louis Kay's lab indicating a possibility. But I mean, I think there's no real reason that it has to be double capped. I think those which are double capped, this is a pretty transient situation, and it's a minority. So the normal state, it's single capped and substrate is fed in from one side. And then, I mean, it might be very similar to the situation, let's say, with Groviel, where also, I mean, things alternate between the two ends. So I think that the fact everybody tried, I mean, in the biochemical preparations to optimize the yield of double-capped 26S, as judged from native gels. And I think that led to a somewhat misleading concept.
Okay, thank you very much again. I think we need to move to the next speaker. Thank you again, Professor Baumeister, for this. So, next speaker is Professor Bolognesi. I do not think he really needs a big introduction. He's very well known. He's a top-class X-ray crystallography. By the way, he has been my tutor, my mentoring for my PhD, so there is a conflict of interest, which I declare publicly here. And so there is also a friendship relation with Martino, and we're really, really looking for his talk. Which is already mentioned yesterday, stimulated curiosity by Professor Ada Ionat about, which is the title, Faeces Infection Diseases Through Structural Vaccinology. Thank you very much, Martino.
Okay, thank you, Menico. Thank you, everybody, for being here, for the opportunity to present these data. Actually, after seeing these beautiful complexes, I think I will present a small molecule crystallography, but let's see how it works. So, and also my talk will be essentially perhaps a methodological considerations into this field. So, yeah. It doesn't move on. Okay, schiacciare molto. Okay. Sorry, just a few slides to introduce the issue of infectious diseases. I'm not going to comment what we heard in last year about the Ebola outbreaks. I just want to bring your attention to this bar over here. This is a histogram, in fact. This is the number of infected or dead people through the end of August this year, and these are the size of outbreaks, of Ebola outbreaks in the previous year. So you see, this was a major outbreak. We are also we have faced in the past other outbreaks. We are still discussing whether this MERS threat, which is conveying viruses from camels, certainly to people, is a serious threat. It infected a few people in the Middle East and a couple of people in the United States. And again, to move on. The last 30 years, so the outbreaks of several viral and, in fact, even bacterial diseases, as we shall see later, to the size of about 40 new diagnosed infections over this time. So never in the history of man that we know, we saw so many infectious diseases emerging in such a short period of time. And this is related to the change in our habits, in our customs. For example, certainly there has been travel, international travel, which is contributing a lot to the spread of infectious diseases, but also climate change, changes in agriculture, in the use of medical devices in the food industry, but certainly drug resistance is playing a dramatic role in this whole picture. I also want to show you this picture, just to wind down my introduction. Since although this is taken from a paper in about 3 years ago, it is showing some of the society features which are facing these days, like failing governance, large-scale population dislocation, conflicts, and so we are seeing that all these things which have their roots into in deeper societal factors are giving rise to the diffusion of infectious diseases. And this is the last introductory slide, which I think is very, it speaks by itself. These are the mortality curves calculated for historical periods like a Neolithic and Paleolithic, but also dated on written records where you see that our ancestors 5,000 years ago were living approximately 25, 30 years, as much as people in Breslau in 1690. And you also see that Liverpool, about 200 years ago, let's say, was not very different from what the Neolithic man was doing and was surviving in those periods of time. Also, we see that there are some parts of the world which are not doing, even nowadays, much better than the Paleolithic man. On the other hand, the red curve here shows contemporary UK, and you see that this dramatic increase in the mean life is essentially took place over the 20th century, when new hygiene, new medicine, and essentially new drugs and vaccination came into play, and so helped men survive much longer. This is essentially a number, the reasons why our lab in Milano, structural biology lab in Milano, is devoting, I would say, almost the whole of our activity to the discovery or to the study of therapeutics which can be used in different fields of medicine and therapy. So we have a research line which is dealing with antivirals. We have a research line which is dealing with pro-apoptotic molecules. We may turn, given recent results, into designing on discovering drugs which are related to epigenetic diseases or degenerative processes. And we are, the topic of my talk today, we are focusing on the application of structural biology to the design of vaccine. Of course, when we talk about vaccination, we cannot help mentioning the roots of the basic ideas which date back to the times of Louis Pasteur, who proposed once you identify the pathogen which is causing the disease, you isolate, inactivate the pathogen, and inject it to elicit an immune response in our organism. So this was the original idea, which during last century gave rise to new approaches to the design of vaccines. So from live attenuated vaccines and vectors, we moved into, people moved into killed vaccines, virus-like particles, and then with the advent of recombinant proteins in the subunit vaccines, conjugate vaccines, and so on and so forth. The big progress came obviously when genomics came into play. You see in the upper part of this slide, you see the common, the traditional or slightly modified way of producing a vaccine, starting from the pathogen, selecting in some way antigens, then testing these antigens against covalescent sera, testing them for immunogenicity, purify, characterize, and identify fully the antigens, maybe modify the antigens and go through this circle several times up to the time when you decide that you can clone the gene for a given antigen, and then move it to testing in animal models and then develop a vaccine. So this whole process is rather slow. It takes between 5 and 15 years maybe, and has been replaced say around the year 2000 by the concept of reverse vaccinology, which is shown here, essentially just to remind you basic concepts. Starting from the genome, you can select through bioinformatics and computational biology the candidate antigens, such I'm talking about protein antigens, which are essentially cell surface proteins, which can be identified, which can be produced in numbers which are compatible with a medium or high throughput expression system, and then can be tested all together in animal models, and then enter the vaccine development pipeline, as I mentioned earlier. So this whole process is much faster than the previous one by a factor of 5 approximately. So building on this concept, then came structural biology. Structural biology, as we shall see in a minute, allows us to focus even more. So without, let's say, once you have identified reactive antigens, you can go further. You can identify regions in the protein antigens, which are really they really contain the epitopes, which are the antigenic determinant of the protein antigen, and then use it for several purposes, as we shall see in a minute. So essentially, what you can do, you can produce antigenic materials which are easy to produce, much more stable than the full original antigen, which can be produced with ease, which can be stored, which can even be coupled into chimeras for cross-protection. Here is an example. Well, I still have this thing, but I don't know how to get rid of this. In any case, it's not a big problem. Sì, sì, c'è. No, in any case, it's not a big deal. Yeah, okay. We keep going. Fine. So the idea here is that we have different variants of the same pathogen which display slightly modified antigens. So in principle, if you want to reach a total immunity against the different serotypes, you have to inject 3 different antigens in the same animal. And maybe these antigens are even big. So essentially, these first 3 steps that you see here are the steps of reverse vaccinology: identification of the antigens, production of recombinant antigens, and injection into the animal. In fact, if you move into structural vaccinology, with methods that I'm going to show you in a minute, you may find out that antigen number 1 is actually reactive, immunoreactive, because of this domain of the protein shown in red. Antigen 2 is reactive for this green domain, and so forth and so on and so forth. So eventually what you can do is edit these antigens into a single polyprotein, which is a hybrid vaccine for total protection against the different serotypes. The example for this is shown here. This is an old study that we published years ago together with the Novartis group in Siena, and which produced, as you shall see, this chimeric antigen, so against group B streptococcus. The idea here is that the Novartis group found that this component of the pili of the bacterium were the antigenic, were essentially the antigen, and they spotted that a given region within this protein was containing the epitopes, which were immunoreactive. The crystal structure of this protein showed that for, I would say, sheer luck, but I mean certainly luck helps. The antigenic region was completely contained in this little domain D3 that you see here in this circle. So since there were 6 serotypes of this streptococcus, the idea was to produce in recombinant form the D3 domain from the different serotypes, link them together in a chimeric protein linked by very simple amino acid linkers, and produce this as a single antigen, which is protective, and in fact it turned out to be protective in animal models against all the serotypes of this streptococcus. So you see, there is flexibility and there is potential in the use, in the coupling of structural information to what is produced by reverse vaccinology. So in the last 4 or 5 years, our group became interested in developing further these methods, and we used melioidosis, but Burkholderia pseudomallei, as a target for the design of new vaccine components. What is Burkholderia pseudomallei? Burkholderia pseudomallei is a gram-negative bacterium which is widespread in the tropical regions, particularly in Thailand, Vietnam, and northern Australia, tropical India, and so on and so forth. There have been a couple of cases also in Italy in the last few years. It is the etiological agent of melioidosis, which is a disease which is essentially starting with lung infection, and which lead to pneumonia, septicemia, abscesses, and go on and so forth. In these tropical countries, in the poorest regions, it can come to a level of about 60% mortality, in fact. It is also a bioterroristic agent classified according to the international regulations. So it shows resistance to antibiotics. So it was really a case for the development of a vaccine. Here is just a few summary slides saying that the disease can show as acute, chronic, as recurrent. It is often a recurrent disease. It can even be latent. Here is an experiment on mice. According to the dose, it can lead to death over a matter of a couple of days, or it can lead to death later on according to the dose that is. And it is also often misdiagnosed as tuberculosis because of several symptoms which are similar. So our approach to the search for new antigens in Burkholderia pseudomallei was based on a network of 6 or 7 groups in Italy and in Europe, which essentially was based on this approach. So in the red line over here, you see that the activity was devoted to the search of antigens. So the search of antigens was done through bioinformatics methods, transcriptomics, proteomics, essentially looking for proteins which were expressed on the surface of the bacterium. And of course, it was also based on literature or existing literature on proteins of this pathogen. So once the antigens were identified, they were produced in a medium throughput approach. If you can read some numbers here, this is an old slide. The numbers that you see here should be approximately multiplied by a factor of 2. So we expressed, we constructed about 100 different antigens. Not all of them were expressed, but we are in the order of 80 probably. These were used for production of immune sera or tested in, as we shall see in a minute, against sera from patients. They were crystallized and their structures were studied either by X-ray crystallography or NMR. The 3D structure was determined, and then this is probably the key point in the whole pipeline. The three-dimensional structures were used for the prediction of, as we shall see, B-cell epitopes. And these then were synthesized as peptides and then sent back to test immunological testing for their effective capacity to induce an immune response and to protect animals. This is just a slide which certainly you cannot neither read nor memorize, showing essentially the pipeline, cloning, expressing, purifying, crystallizing X-ray structures, immunosera productions, and so on. This is part of the whole story. These are some of the antigen structures which have been produced, and I'm going to show you 2 or 3 examples from these, showing how the three-dimensional structure helped us to find the epitopes and produce peptides which were active. Okay, the first case is, let's call it PAL-B, this protein, which is a peptidoglycan-associated lipoprotein, which is conserved in 28 Burkholderia genomes analyzed, so it's a good candidate for vaccinology. It was known from the literature that it provides 50% protection in mice immunization experiments. So we went through the crystal structure analysis, conventional stuff. It's a small protein, 170 residues. It's a trimer in solution, and we then used the crystal structure of the monomer to predict epitopes. And I will spend a few minutes on this methodological approach, which brings in computational biology. So essentially, we used in collaboration with Giorgio Colombo at CNR in Milano, we used, actually Giorgio used 2 prediction methods which predict regions of the protein which are likely to be epitopes in this antigen. So the first method, matrix of local coupling energies, essentially looks for, and I will show it in the next slide, essentially looks for regions of the molecule which are relatively free of interaction with the core of the protein. The second approach, electrostatic desolvation profiles, looks for regions in the protein which are on the surface and which can be desolvated rather easily. And I'm showing this in the 2 slides. Here is the matrix of local energy coupling, which is essentially calculated this way. The top region, the top figure is the contact map. So you have the sequences of the 2 proteins, and you calculate physical interactions really between contacts between residues, as has been used for years now. This is an energy map which comes after some MD calculation and so on. If you make a convolution between these 2 matrices, you find out regions which are in contact, as shown here, but which are relatively devoid of local energetic interactions. And so these are likely to be flexible regions on the surface of the protein, which are likely to become epitopes. Desolvation energy. Here is an example. Here is, I think, catepsin with a protein inhibitor, so, which is known to assemble into a protease inhibitor complex. As you can see, the interaction surface between these two is red. Red is not electrostatic here. We are just coding the dissolution energy. The dissolution energy is shown here essentially through simulations with water probes and a neutral probe on the surface of the protein. You can calculate how easily you can remove water from the protein surface. And in fact, it is intuitive that if you want to bind one protein to another, the removal of water molecule can be an obstacle to the assembling of the complex. So if you can remove water, dissolvate the interacting surfaces easily, this is likely to be a protein-protein interaction region, as is the case of epitopes with antibodies that we are searching. So applying this to this target, we found, Giorgio found 3 potential epitope regions, which are mapped here on the sequence. These were also, which are mapped here again on the sequence, which were also mapped through, I really have not so much time, through an experimental approach which involves partial digestion with the protease and capture with polyclonal antibodies and identification through mass spectrometry. But this information led us to synthesize 3 peptides which were produced in vitro and which were tested then against sera from seronegative patients, melioidosis seronegative patients, melioidosis seropositive patients, and recovery patients. And we see that in this case, which was the first one that we studied, this is the whole antigen. This is 2 peptides which were not reactive, and a 3rd peptide, epitope 3, as we called, showed and improved the reactivity, allowed to distinguish between seropositive and seronegative patients. So it was the first example that showed that our method was potentially working. In fact, the same peptide was able to raise antibodies which led to agglutination of Burkholderia pseudomallei even better than the whole antigen. And as shown here, I have really very little time to go into details, but I think you can appreciate this. It also led to phagocytosis and to oxidative burst in human neutrophils, as shown by these data that I'm skipping for brevity. So the peptide was working. The same results were obtained with a different antigen in our pipeline that you see here, and also going very quickly, I'm showing that the peptides produced by the same approach were reactive against sera from seropositive and recovery patients. And the last one I'm going to show, 3 slides. It's another antigen which was isolated. This is a component. This is flagellin from the flagella of the bacterium. Here again, we had a beautiful 1.3 angstrom structure. This is not really relevant for immunogenicity, but we enjoyed it. And in fact, in this case, we were able to predict something like 6 different peptides, 2 of which were particularly well recognized by sera, this one and this one, as B-cell epitopes, 2 of which were not recognized as B-cell epitopes, as you can see here, but actually were recognized as T-cell epitopes. In fact, in the case of this antigen, we we're applying, this is the last one we have been studying, we were applying additional computational methods which were also relevant for the prediction of T-cell epitopes. And in fact, even these peptides were able to induce phagocytosis, oxidative burst, and bacterial killing and offered even protection in animal testing. So, to cut a long story short, my examples have shown you a methodological approach to vaccinology. We are confident that these methods in a short time will develop into something which is reliable and in fact, it can be applied to any pathogen. I showed you the case of Burkholderia pseudomallei, but we can apply it to any pathogen as long as we can produce recombinant protein and study three-dimensional structures. So in a way, several of the steps that are listed here are already in our hands, but certainly not all of them. And the conclusion, in my opinion, is that we are moving in this direction and we are producing a method which will be of general applicability, but there are basic questions like what is the molecular nature of immunodominant epitopes, or why some antigen are strongly protective and others are not. And I think that the answer to this problem is rooted in basic science. So we need basic science to explain questions that are rooted in the molecular structures, in the dynamics, and in first principles of protein structure. So this is the message that I wanted to convey. I just want to acknowledge two people in the lab, Louise Gorley and Patricia Lasso, who were responsible for all the data, the structural data that you have seen. Giorgio Colombo in Milano, again, CNR, who is the group leader for the computational biology aspects. Xavier Daura in Spain for the epitope mapping through mass spec. And of course, our connection, Ganjana Lertme, okay, I'll leave you to read it because I cannot pronounce it at all, for sera testing against patient sera, and also University of Exeter, Rick Tidball, for part of the in vivo experiments and animal model experiments that we have been running. And thank you for your attention.
So thank you very much, Martino. Again, another example of multidisciplinary approach to use structural biology and computational biology. So certainly time for a couple of questions to Martino, if there are any. Andrea? Again,
Martino, thank you for your presentation. Is there any lesson from the negative result of the prediction of epitope?
Yeah, in some cases, yes. I had no time to go through it. For example, here, you see this is totally non-reactive. It was predicted based on sequence methods. And in fact, the structural methods show that this can't be an epitope because in the assembled pilum is part of an interface not accessible. So there are, but not always, not always.
Is there? Yeah.
Thank you, Martino. An effective immune response must involve also the way antigens are presented in vivo. Yeah. Do your approach contribute something to this second part, I mean, antigen presentation?
No, not yet. We are moving into the idea of T-cell antigens, or epitopes even better, so which would mean presentation through the MHC complex Oh, sorry, only recently. So we started with, from B-cell epitopes, which are essentially protein-protein interaction in a way. So epitope-antibody interaction, because this is certainly much closer to our knowledge of protein structure and protein activities. But we are, as I said, in this last case, we are moving into even the prediction of these, but certainly as I said, I'm not an immunologist. Immunology is extremely complex, in my opinion. So we try to go step by step and produce something which we can trust on ourselves.
So if no more questions, immunology, I think they are 10 years ahead of us, but they even don't know that.
Sorry,
so immunologists, they are 10 years ahead of us, but they don't know, so they cannot communicate this.
Ahead of us because they start from the macroscopic.
Exactly.
So we have to meet.
So if there is no more questions, thank you again. Okay. Martino, we move to the poster selection.
Okay. So we now move to the next two speakers of this session. And the first one is Stefano Ricagno from the University of Milano. And he will speak about Embelin binds to human neuroserpin and impairs its pathologic polymerization.
Yeah, okay, probably closer to my mouth. Okay, right. So, good morning, everyone. Before starting my presentation, I want to thank the organizers for giving me the opportunity to present my data. And I also want to thank the organizers that yesterday at dinner time told me that my presentation was supposed to be 20 minutes instead of 10 minutes, right, but it's going to be shorter than 20 minutes. So, okay, so I thought maybe I could repeat twice the presentation, or maybe I can provide some more data, and that's what I'm going to do at the end. So I'm going to talk about neuroserpin, its toxic polymerization, and the action on these polymers done by this small molecule that is called Embelin. So a little bit of introduction. Human neuroserpin, it's a protein member of a large family of proteins called serpin family. The most part of these proteins are serine protease inhibitors. And as the name goes, neuroserpin is mainly expressed in the nervous system, where it's specifically inhibiting the tissue plasminogen activator, TPA. We are studying this protein because 7 mutants of neuroserpin has been found to be responsible for a rare genetic disease named FANIB. The acronym is FANIB, but it's actually familial encephalopathy with neuroserpin inclusion bodies. The symptoms are early onset dementia, epilepsy, and other neurological disorders. The polymers have been found in all the patients accumulated in brain. And actually, from the biochemical point of view, it's very nice because more unstable is the mutant, more severe are the symptoms of the disease. So there is a very good correlation between the biochemistry and the symptoms. Just to give you an overview about how this protein works, can we lower the light? No, we can't. All right. Okay. So for the inhibition mechanism is the following. Here we have the serpin. Probably don't see it very well. Probably I should have made it lighter. But here we have a long exposed loop. That is used as a bait for the serine protease. The serine protease binds this loop, cleaves the loop, And there is this big conformational change leading to this new conformation, the cleaved conformation, serpin cleaved conformation, where this loop becomes a new beta strand inserted in this large beta sheet here in the middle of the protein. So there are obviously at least two, but actually three monomeric conformation of neuroserpin. The first one is the native, as I just showed you, where this loop is exposed and flexible. The second is the cleaved conformation, where the loop is indeed cleaved as is inserted. These are two structures, neuroserpin structure. This is not a neuroserpin structure, it's another serpin. But the third is the latent conformation, where you see the conformation is very similar to cleaved, but in fact, the loop is not cleaved. So the uncleaved loop is inserted in the protein. So these two conformations are instrumental for the physiological functioning of the protein. This is one of the two misfolding conformation. The second one, the really dangerous guy, is the polymeric conformation. There are just two competing models that are fighting a lot to be the right ones. I'm not going to get into that. Just want to stress that whatever the polymer is, we are not talking about amyloid fibrils, cross-beta structures, but these polymers are native-like polymers, where the protomers have fold very similar to the native conformation. Right. So to summarize the complex energy or conformational landscape, if you like, of serpins and of neuroserpin, we have the native conformation here that can be cleaved to become the cleaved conformation and becoming much more stable, while the native is a metastable conformation. And this part here on your left is the functional folding, while on your right, the metastable can also overcome this energetic barrier and becoming latent form or polymeric. Especially the mutants are destabilized in the native form. This energy barrier becomes lower, and that's why the mutants are spontaneously forming latent and polymeric conformers. Right. Working with the wild-type neuroserpin is much easier than with the mutants, so we've been working always with the native, but we can actually form very easily polymeric and latent form by incubating overnight at 45 degrees the neuroserpin. And if you see here in this gel filtration, this is the continuous line is the native, the dashed line is the polymer that is always eluted at the dead volume of the column. And here is actually very small, but there is also some monomeric that is the latent. But let's concentrate on this is the native, and at the dead end is the polymer. Among many things we've done to characterize polymer and latent conformers, I'm just telling you a couple of things about the chemical unfolding. So native and latent, so the monomeric conformations have this melting concentration, 2.9, 3.5 molar guanidinium hydrochloride, while the polymer has the polymerization concentration, so it's not unfolding. It's just when I see in a gel filtration experiment when the polymer becomes monomer. That is 2.8. That is resembling rather closely the melting concentration of latent, or is close to the native and to the latent. So probably under this condition, the polymerization and the unfolding are probably either concomitant or anyway very close, very close. Okay, I hope I convinced you that the polymer is very stable because, well, it is very stable. So obviously, one of the most interesting things that we could do is to find some kind of compound that could actually prevent the polymerization or destroy the polymers. Reading literature, we saw that that Embelin, that is this compound shown here, binds to another, was shown to bind another serpin, pi1, and binding specifically and binding and having some effects in the enzymological inhibition. So nothing to do with the polymerization. But since there are not many compounds, actually there are no compounds that are binding to serpins, we started from this because we thought there might be some interest in that. And obviously we are interested in polymers, so we tested this Embelin with serpin polymerization. So the first thing was to incubate the serpin, the neuroserpin, at 45 degrees overnight. I remind you that without Embelin, this is what we get. So in the dashed line is the very big polymers and some latent form. While in presence of Embelin, we were getting something different. We were getting some oligomers. You probably think it's not very exciting, but typically we never get anything smaller than the super huge polymers. So that was already a big surprise. We incubate the neuroserpin at different, you don't see that, but at different temperatures, realizing that the end of the process is indeed the formation of these intermediate-sized oligomers. So this was already something interesting. We wanted to see what was happening when we were adding and we were incubating Embelin with preformed polymers to see what was happening. And to our joy, we incubated at different temperatures, so the kinetics of the process is different, but the message is clear, so that incubating preformed polymer with Embelin overnight at 45 degrees, we get the degradation of the big polymers to something smaller, reminiscent than to the oligomers I showed you before. So with this kind of 1.5 millimolar Embelin solution, we could get pretty much what we got before with 2.8 guanidinium hydrochloride. So that was, in my view, a nice result. We tried to monitor the serpin polymerization using different experiments. In the serpin world, you have to do native gels, otherwise referees are not happy. So that's what we did. Without Embelin, we see that the native becomes by time bigger and bigger as to form the big polymers, as we saw by GF. In presence of Embelin, by time we never get to very big polymers, but we stay to intermediate-sized oligomers, as we saw in the GF experiments. By fluorescence, we did the same thing, comparing the polymerization in presence or in absence of Embelin. These lines are in absence of Embelin, a different concentration. And we see that in presence of Embelin, something different happens. The fluorescence is, in fact, much lower. This raised a few questions that we wanted to address. Number one, what are these oligomers? Are these oligomers folded, or we are dealing with unfolded, uninteresting, I would say, protein? No, we do have folded oligomers. Here we have CD spectra of native neuroserpin, polymeric neuroserpin before and after Embelin incubation. And there are some minor conformational changes, but typically we can say that the overall fold is kept. Then we used 4 antibodies specific against serpin polymers, and we tested against our oligomers. As we can see here in the black lines are the affinity against the polymers that you can see is very high for all four of them. While here in red or orange is the affinity against our oligomers. That is very low, as low as the affinity against the native. So that means that what these four antibodies are telling us is that the polymers are not just smaller, that the oligomers are not just smaller polymers, but they are something different. Then we wanted to say, okay, these oligomers, do they bind Embelin, or the effect is some kind of unspecific effect that Embelin is exerting in solution? No, there is a binding. We tested all four conformers, latent polymer, native polymer, latent and cleaved form. I'm not going to get into the details, but they coelute in gel filtration, saying that there is a stable binding. Is this binding specific, or we have a binding somewhere on the surface with random stoichiometry? So that was also a question that we wanted to address, and we do have a specific complex with a molar stoichiometry of 1:1 that we've done by mass spec under native condition, where we could actually observe the monomeric complex 1:1, and also analyzing dimer, trimer, and tetramers, we could see the 2x2, the 3x3, and the 4x4. And as you can see here in the eye panel, all these are folded protein. So there is no unfolding unspecific material in these experiments. Right. Last experiment that we've done was to see how neuroserpin is behaving once it has interacted with Embelin as a protease inhibitor. So here we are testing TPA activity in presence of native neuroserpin. We have lower activity as expected because neuroserpin is an inhibitor. While when we added the oligomers, then we could see that there was no decreased activity as when the protein, the TPA, is alone. And here in these gels, we could see that the neuroserpin oligomers are not inhibiting, but they become actually substrate. So they are cleaved by TPA. So to conclude all this, I hope that I convince you that Embelin displays an antipolymer, actually a dual antipolymer effect, both by preventing polymerization and by degrading the polymers that are already formed. These oligomers with presence of Embelin are TPA substrate and not inhibitors, and this also could also have some application that I didn't mention for sake of brevity. So what I want to conclude with is that molecules with Embelin properties have great potential as hits against neuroserpin polymers and therefore against FANIB. We need to have a lot of we need to do a lot of work, because for now Embelin is very toxic compound, so we could not do any experiment in cell. That is a weak point of the work, but we need to find a version of Embelin that is less toxic. Acknowledgement. A lot of people did a lot of great work. At University of Milan, main mention in Fabio, Giorgia, Alberto Barbirolli here in the audience, Rosaria Russo. Then at University La Sapienza di Roma, Elena's group did the West blots. Some biophysics was done in the Institute of Biophysics in Palermo at the CNR. And the University of Bicocca in Milan, they did the mass spec analysis. And I thank you for your attention.
Oh, perfect. Thank you very much. So we have time for 1 or 2 questions.
Do you have some idea on the mechanism by which those native protein polymers are toxic? So, because if you imagine that this partially disassembling the complex will ameliorate the disease, it means that in your mind the oligomers are less toxic than the full fibers.
Yes, that is the typical question in the misfolding world. So that is not clear. Is not clear. Is obviously has nothing to do with the real misfolding, where the oligomers are typically hydrophobic patches, problem with membranes, etc. Here, everything is folded, nicely soluble, etc. What the people. The ultimate answer is that it's not clear, but the accumulation is very often in the ER. It's partially in the ER, partially outside the cell. The accumulation in the ER is actually clocking the ER, making its functionality very, very, very difficult. So probably some of the toxicity comes from that.
Yes, one, yeah, that means.
In view of possible applications, can you say something about the concentrations to be used for Embelin concentrations?
Yes, in all these experiments, just to standardize the conditions, we used 1.5 millimolar. That is saturated because Embelin, among many other problems, is also very little soluble. We consider this Embelin as just a starting point because not for neuroserpin, not for any of the other proteins, there is a compound that has been published to have antipolymer effect. So this is very new. However, Embelin is very far from being an ideal compound because it's toxic, it's insoluble, and we haven't really done any affinity experiment to see what is the relevant concentration.
Okay, thank you very much. I'm sorry, but we have to cut this discussion and move. Thank you again. Move to the next speaker, who is Luca Federici from the University of Chieti Pescara, and he will speak about mapping nucleophosmin interaction to the targeted treatment of acute myeloid leukemia.
Okay. First of all, let me thank the organizers, and particularly Manico, for this invitation. And today, I'll try to give you just an overview of what we are doing about this protein called nucleophosmin and the possibility to use this protein as a target for the treatment of acute myeloid leukemia. So nucleophosmin is a very abundant protein, which mainly resides in nucleoli, but it's able to shuttle between nucleoli and the nucleoplasm and the cytoplasm to fulfill its many functions. You can see some of the function. It is essential for ribosome maturation and export, chaperone activity, and cell cycle control, and apoptotic response to stress and oncogenic stimuli. And this is done through the interaction with both nucleic acids, both RNA and DNA, and a plethora of protein. And if you look at the protein, we don't have the complete structure, but just structures of the isolated domain. You can see that we have an N-terminal domain, which is a beta sandwich, which assembles in pentamers, and then two pentamers assemble to form a decamer. And this is the domain responsible for the majority of protein-protein interactions. Then we have a central unstructured domain, really acidic, which is essential for histone binding and assembly. And finally, we have a DNA-RNA binding domain at the N-terminus, which you see is this short and small 3-helix bundle. Then we have a number of signals which dictate the movement of the protein between the nucleoli and the cytoplasm. So we have nuclear export signals, nuclear localization signal, and the nucleolar localization signal, which is at the C-terminal domain. So, Nucleophosmin gene is the most frequently mutated in acute myeloid leukemia. We're talking about 35% of the patients. Mutations are many, but they are somewhat similar between each other. First, they are always heterozygous. So you have one normal allele, and in the blast you have both the normal protein and the mutated one. And if you look at the mutation A, which is by far the most common, you can see that at the last exon of the gene, so the extreme end of the protein, you have an insertion of 4 bases. This changes the reading frame, and at the protein level, you will have a different sequence at the very extreme C-terminus of the protein, and I show you that you lose both these tryptophans that were readily recognized as the nucleolar localization signal of the protein. But together with this, there is also the appearance of a new nuclear export signal. So what happens to the protein? The protein, which is nucleolar, the majority of the protein is nuclear, as I told you before, goes to the cytoplasm because it loses affinity for nucleoli, and because of this new nuclear export signal, it goes in the cytoplasm. And I can show you this. These are data from my lab. And you can see that this is a leukemia cell line with wild-type nucleophosmin at both alleles. And you can see that the protein is nucleolar. This is a leukemia cell line where you have the mutation A, heterozygous mutation A, and you can see that the protein, in the majority of the protein, is in the cytoplasm. So, as I told you, this is the most common mutation in leukemia. There are a number of evidences that also say that this is the initiating mutation of this kind of disease. And therefore, nucleophosmin is considered as an ideal target for the treatment of this kind of leukemia. The problem is how to treat it. And one hypothesis, which there are many hypotheses, but I will focus only on this one today. This was proposed by Brunangelo Falini, who is also the discoverer of the mutation, is the so-called Nucleophosmin nucleolar starvation hypothesis. The idea is that the majority of the protein in the blasts is in the cytosol, no longer in the nucleoli. And this is I want to stress not only the mutated protein, but also the majority of the wild-type protein, because the wild-type protein oligomerizes with the mutated one, it will go in the cytosol all the same. So in nucleoli, you have only a small amount of nucleophosmin, and the idea of Professor Falini is that if we could find a drug capable of selectively delocalizing wild-type nucleophosmin, the residual wild-type nucleophosmin from nucleoli, since the amount of this protein in the blast is much less than in the normal cells, we should be able to cause nuclear stress, impair the ribogenesis, and death of the blast by apoptosis. In order to do that, we first, in the first place, have to understand why nucleophosmin is in the nucleoli. And since mutations localize at the C-terminal domain of the protein, I just will focus on this domain in the next slides. As I told you before, the domain is responsible for DNA binding and also RNA binding. It was already known by the 90s that it has a strong preference for single strand in the samples, no sequence requirements, and the interaction was, as I told you before, mapped to the C-terminal domain. When we started this investigation, there was only one single sequence, one specific sequence that was known to be bound by nucleophosmin in vivo. And this is this sequence that by Watson and Crick pairing is predicted to fold as an hairpin, and which is found in the SOD2 promoter. And by binding this promoter, MPN1 participates to a network of interaction that activates the transcription of the gene. So we started this investigation, this in vitro investigation with this oligo, and we cloned two different domains of the protein. A shorter domain, which is just the terminal 3-helix bundle, which is the minimal folding unit of the C-terminal domain, but also a longer one, which comprised, I'm sorry for the slide, which comprised a tail enriched in positive residues. We used SPR for assessing those interactions, and you can see that we have pretty good affinity if we use the longer domain, while the affinity dramatically decreases if we use the shorter domain. So the tail appears to be important for recognition. Then we started to play a bit with the structure of the hairpin, just to make some structure and function relationships. And if you see here that the physiologic substrate has 10 Gs in the loop, what happens if we replace these 10 Gs with Ts? You see that we have a dramatic loss of affinity. Also, the number of Gs is important, because if we reduce the number to 5, we also have a loss of affinity. We confirmed that nucleophosmin is able to bind with low affinity also an oligo with no specific sequence. This is a poly-T. What surprised us was the result obtained with the poly-G oligo, the linear poly-G oligo, where you see we have an affinity which is comparable to that we have with the physiologic substrate. So at this point, we can tell that so a larger domain is necessary for high affinity recognition. So we need the tail. The G-loop 3D structure appear to be important because substituting the Gs with the Ts, but also reducing that number, reduces the affinity. But why, above all, why a poly-G oligo is recognized with the comparable affinity? We came out to understand that the poly-G oligo is not linear as we thought it would be. Instead, it is folded as a G quadruplex. You may know about G quadruplex assembly. Basically, you need to have in the same oligo 4 consecutive stretches of at least 3 Gs separated by loops where you can have any kind of base. And so if a poly-G oligo is long enough to fulfill this folding rule, it will fold as a quadruplex, in this case as an ensemble of different quadruplexes. I won't show you the data, but we collected them. So maybe nucleophosmin displays a higher affinity for G quadruplex structures. We used, to prove this, we used a well-known quadruplex, which is the one at the CMYC promoter. This is very well characterized in terms of structure, and it also forms in vivo. And once again, you can see that this is bound with good affinity by the longer domain and with less good affinity by the shorter domain. So, retrospectively, we went back to the original oligo, the physiologic one at the SOD2 promoter, and we could demonstrate that the G10 loop, which we thought would be an hairpin, instead, at least in vitro, it's a quadruplex. It fulfills the folding rule, and we accumulated many proofs of this, but I think that just simple static CD spectra are enough to describe that it is identical to, its behavior is identical to the simic oligo. And you can see that in the presence of monovalent cations, you have an increase in intensity of 262 and the formation of this through at 240, and both these features are diagnostic of G quadruplex formation. So, the next question is, which is the role of the tail? The tail is essential for high affinity binding with all the oligos I showed you, whatever the structure. And, but the tail is also predicted to be natively unstructured. So we asked whether this is true and whether it does fold upon DNA binding. Since we couldn't crystallize this object, we collaborated with a group of Lucia Banci at Florence, and we determined the structure of the longer domain, which was unknown, and you can see that the tail is not structured, and it does not have any propensity to assume a secondary structure according to NMR data. Then we studied the complex with the simic G quadruplex. We used the shorter version of the quadruplex, basically because this was already known in structure, and this facilitated our task. What you can see here is that the quadruplex is recognized in a small groove, which is formed between helices 1 and 2. And what is important here, always regarding the tail, is that we could not observe any relevant chemical shift variation in the tail. So if you look at the structural model, the tail does not seem to participate to the final complex. This seems a contradiction with what I've told you before, because the tail is always essential for high affinity recognition. So we did some mutational studies to clarify this issue. Single mutation, you can see that the tail is positively charged. We have 5 lysines. We mutated each of them. Single mutations have little or no effect, but if you start mutating them in couples or in triplets, you can see that you can reach dissociation constants which are comparable with the absence of the tail. Most important, if you look at this isoaffinity plot, I plot the k-on for each mutant as a function of k-off. So this is the wild type. And if you look at the majority of the mutants, you have both a decrease in the k-on, but also an increase in the k-off. This means, in our view, that the tail not only function through electrostatic interactions that facilitate the formation of the encounter complex in a fly casting scenario, but also some way they contribute to stabilize the final complex. To corroborate this, we performed some molecular dynamics simulation, and this is the protein, this is the quadruplex, and this is the tail. What you see in red is the contact surface between the two. And you see that the tail transiently contacts the quadruplex. Now, in a second, you will see the tail going back. Takes a bit too long. Okay, now it's back. And then you will see the tail going again, embracing the quadruplex. So, in our view, these are transient interactions. So we cannot spot them in the NMR time scale, but they nevertheless contribute to stabilize the final complex. So, while we were doing these biophysical studies, we were also wondering, but does this nucleophosmin G quadruplex interaction has any relevance, any physiologic relevance? And we realized that nucleoli are organized around tandem arrays of ribosomal DNA genes. And this is the ribosomal DNA genes. And we also realized that this ribosomal DNA gene is plenty of putative G quadruplex sequences. Not only this, the sequences are in the non-template strand, so that you will also find the G quadruplexes in the nascent RNA. So we first asked whether nucleophosmin binds these G quadruplexes, and you can see here that nucleophosmin actually binds them both in vitro, but most importantly, but most importantly in vivo. You can see from these chromatin immunoprecipitation experiments, these are all G-quadruplex sequences in the rDNA genes, while this is a negative control. This is a non-G-quadruplex sequence at the beginning, at the end of the promoter and the beginning of the transcription start site. So, nucleophosmin binds G-quadruplex sequences at nucleoli, and if we come back to the protein and the mutated protein, I show you again what happens when you have the leukemic mutation. It happens that this part of the protein is different, and you lose both the tryptophans. In terms of protein, it means that this is the wild-type protein, this is the mutated one, that while the wild-type protein is folded as a 3-helix bundle, the mutated one is completely unfolded. You can also see this in the thermal denaturation profile. And from the functional point of view, it is not surprising to observe that the wild-type protein binds the G-quadruplexes, while the mutated protein does not. So we thought maybe the protein is in nucleoli because in nucleoli it binds nucleic acids. And in order to demonstrate this, indirectly, I have to say, we used a very well-known G-quadruplex ligand. This is a porphyrin. You can see the porphyrin here. And you can see in this SPR experiment that the porphyrin is able to compete with the quadruplex for the protein binding. So this can be used as a tool to demonstrate that nucleophosmin can be displaced from nucleoli. And this is what we did here. You can see this is nucleophosmin, the nucleoli. If we treat it with TMP-YP4, you will see that the protein starts to detach from nucleoli at 12 hours. It's almost completely nucleoplasmid at 24 hours, and then the cells will start to die. So, we believe this is a proof of concept. It is possible to dislocate MPN1 from nucleoli by interfering with its DNA binding properties. And if I come back to the initial nucleolar starvation hypothesis, I hope to have convinced you that if we want to find a drug that displaces nucleophosmin from nucleoli, we just have to find a molecule that binds the protein here. Where is the interface with the G-quadruplex or the G-quadruplexes that it binds. And finally, I wish to thank people working in my lab, especially Sara Chiarella and Mimma Franceschini, and above all, Adele Di Matteo, Angelo Gallo and Lucia Banci for the NMR data, Alessandro Arcovito, who is in the audience for SPR data, Stefano Della Longa for the molecular dynamics simulation, and let me also thank my mentor, Professor Maurizio Brunori, for his continuous support in developing this research. And thank you very much.
Thank you very much. We are nearly on time, so we have time for one question.
So, may I start? So thank you very much, Luca. Very fascinating talk. So just a question concerning, it's very nice, you know, the data concerning the affinity of the nucleophosmin and the quadruplex structures, because, you know, there should be a drug modulating this interaction. But coming back to the initial questions you presented, do you think that this affinity is able to modulate the translocation of the protein from the cytosol to the nuclei? And is there possible that the tryptophan's mutations is also able to impair in some way, you know, the interaction with the membranes, I mean nuclear membranes, you know? In that case, the translocation should be such in other proteins modulated by different affinities with different specific membranes. Have you any idea about that?
There are two questions here, if I understood well. So the first question is if it's because of the affinity is not very high, I've seen. It's around 10 micromolar. That yet it is enough to keep the protein in nucleoli. What moves the protein from nucleoli are post-translational modification, which I didn't show you for the sake of time. In particular, acetylation, and acetylation happens, if I come back, acetylation happens in multiple places, both here and here, but also in the tail. When the protein is acetylated, it detaches from nucleoli. The second question is about possible membrane attachment of mutated nucleophosmin. I just guessed that nucleophosmin goes in the cytoplasm because it is recognized by a particular exportin 1, also called the CRM1. And I guess that recognition does not happen at the C-terminal domain of the protein. I showed you that nuclear export signal is at the N-terminal domain of the protein. So when the protein is mutated, it loses affinity for nucleoli, and then it will be recognized readily by CRM1, and then it will go to the cytoplasm. There are no data arguing against the nuclear accumulation of the protein. There are data arguing for membrane accumulation of the protein once it is cytoplasmic, but that is because it interacts with another protein which is called nucleolin. So that's another story.
I think if you don't mind, we can discuss at the coffee break. So I would like to thank all the speakers again, and we are 5 minutes late, so it's good. Thank you very much.